students, my name is Niyati Said and thanks for watching Wikipedia word videos. My topic for the presentation is cell cycle regulation. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproductions are characteristics of cells, indeed of all living organisms. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycle of growth and division allow a single cell to form a structure consisting of millions of cells. Okay? So what is cell cycle? Cell division is very important process in all living organisms. We know that. During the division of a cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Okay. So, what is a cell? Cell is a structural and functional unit of all living organisms. Okay. Cell cycle is a cycle. It has no distinct beginning or ending. Cells are continually entering and exiting the various phases of the cell cycle. Okay. Cell division, as I have told you, that it is a process of the formation of a new cells from a pre-existing cells, that is referred to as cell division. Just see this illustration. Suppose you bought a new cell phone. Okay. Your old or unused phones, you wanted it to be recycled. Okay, that's why you donated to a NGO or you did charity. Okay, benefit is that the environment and the people in need, and this cycle goes on and on and on. That is the same analogy with the cell cycle. Okay, which is a fundamental unit of our body. What is cell cycle? The period in which one cycle or cell division is completed. That is called as cell cycle. It has two phases. Interphase, at which DNA synthesis takes place. Okay. And second is the mitotic phase. In this, at which nuclear division takes place. Okay. Always know that mitotic phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive mitotic phases. Okay, it is significant to note that in the 24-hour average duration of cell cycle of a human cell, division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome. That process is known as karyokinesis, and usually ends with the division of cytoplasm, that is known as cytokinesis. The interphase, though called resting phase, is the time during which the cell is preparing for division by undergoing cell division and cell growth and the DNA replication in an orderly manner. Okay, so Interphase, it consists of three stages. One is G1, that is gap 1. Second is S phase, that is synthesis phase, and the third is the G2 phase, that is known as gap 2 phase. Okay. G1 phase, it corresponds to the interval between mitosis and the initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase, it marks the period during which DNA synthesis or the replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is donated as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2n number of chromosome at G1, even after S phase, the number of the chromosomes remains the same, that is 2n. Okay. In animal cell, during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for the mitosis while cell growth continues. Okay. This is the proper illustration of a cell cycle. This is a cell in the G1 phase, and then it goes into the synthesis phase where the karyokinases takes place or the chromosome duplication takes place. Then it enters into the G2 phase, that is a gap 2 phase, in which cell with the duplicated chromosome moves and forms or divides the cell. Then it comes into the mitotic phase. Okay. And this entire cell cycle is regulated by CDK and cyclin. Okay, CDK is a cyclin-dependent kinase, and the cyclin, they both are the regulators of the cell cycle. Okay. Now, we will be studying regulation of cell cycle in detail. Cell cycle controls play a fundamental role in plant by supporting their largely post-embryonic growth and development. Core regulators of the plant cell cycle. 4 distinct cell cycle phases are generally recognized: G1, S, G2 and M. In G1 and G2 phases, during which cell cycle machinery is prepared to enter the next phase. These gap phases function also as a checkpoints. Okay, checkpoints we will be studying in detail. Okay, among the bulk of cell cycle players, cyclin-dependent kinase, that is CDK, is coupled with the cyclin, play a crucial role in the regulation of cell cycle progression. Okay. Now we will study CDK and cyclin in detail. CDK is a cyclin-dependent kinase. They are the family of protein kinases which was first discovered for their role in regulating the cell cycle. They are also involved in regulating transcription, mRNA processing and differentiation of nerve cells. They are present in all known eukaryotes, and their regulatory function in the cell cycle has been evolutionarily conserved. Okay. In fact, yeast cell can proliferate normally when their CDK gene has been replaced with the homologous human gene. Okay. CDK are relatively small proteins with a molecular weight ranging from say 34 to 40 kilodalton and contain little more than kinase domain. Okay. By definition, a CDK binds a regulatory protein called cyclin. Without cyclin, CDK has a little kinase activity. I repeat, by definition, a CDK binds to a regulatory protein called cyclin, and without cyclin, CDK has a little kinase activity. Only the cyclin CDK complex is an active kinase. Okay. CDK phosphorylate their substrate on serines and threonines. So they are serine-threonine kinase. Okay. And if we talk about plant CDK, then it can be classified into 7 different groups from A to G on the basis of the characteristics, amino acid and cyclic binding motifs. Name of CDK and amino acid cycling binding motif: CDKA, CDKB, CDKF and CDKD, CDKC, CDKE and CDKG. Okay, and these are the active amino acid cycling bonding. motifs. Okay. Among the CDKA plays a pivotal role during the whole cell cycle, being especially important for the transition from G1 to S phase and G2 to M phase. Okay. The later transition G2 to M phase is further regulated by another type of CDK that is exclusively to plants, the CDK-B type. Okay. CDK-D and the plant-specific CDK-F function as a CDK activating kinases. That means they activate AKs. Okay. CDK-G is possibly involved in coupling cell cycle progression with the splicing of transcripts. Transcripts is a mRNA. As we know, in the process of transcription, DNA is transcribed into messenger RNA. Okay. CDKs C and E have no direct participation in the cell cycle. Okay. Now come to the cyclins. Cyclins, as the name cyclin, CYC stems from the transient and the cyclical appearance of these proteins during the cell cycle. Cyclins, you can say, they are the family of proteins that control the progression of cells during the cell cycle by activating CDK enzymes. Okay. Cyclins were originally named because their concentration varies in a cyclical fashion during the cell cycle. Okay. The oscillation of the cyclins, namely fluctuations in cyclin gene expression and destruction by the ubiquitin-mediated proteasome pathway, induce oscillation in the CDK activity to drive the cell cycle. A cyclin forms a complex with the CDK, which begins to activate the CDK, but the complete activation requires phosphorylation as well. Complex formation results in the activation of the CDK active site. Cyclin themselves have no enzymatic activity but have binding sites for some substrates and target the CDKs to specific subcellular locations. Okay. They were discovered by the scientist Timothy Hunt in 1982 while studying the cell cycle of sea urchins. Okay. Most cyclins are characterized by 100 amino acid sequences. They are known as cyclin boxes that is required for the proper binding to the CDKs. Okay. And plant specific feature like post-embryonic development, meristematic growth, or even development alteration in response to environmental fluctuations, plants should have evolved a tightly regulated yet flexible cell cycle machinery, which could explain this high number of cyclin. Okay, these are the cyclin representations: cyclin D1, D3, cyclin E, cyclin A, cyclin B, and cyclin 13 and SIG2. Okay, and these are the CDK protein kinases which these cyclins bind up, and thus cell cycle progresses. Okay, and these are the cyclin levels which it increases or the decreases, and these are the note how they start, at which point they start. Cyclin D1, D3, they start at the G1 phase progression, and cyclin E, they are present at the onset of the S phase, that is synthesis phase. This cyclin A, it helps in the S phase progression. Cyclin A, it goes from synthesis phase to the gap2 phase. Okay, and cyclin B, it helps in the metatic phase progression, and this cyclin 13, it prevents S phase before M phase. Sometime it is seen that S phase moves to the phase. Okay, this is the representation: mitosis, interphase, mitosis, and interphase. Between two successive mitosis phase, there is an interphase phase. Okay, and it is seen that in the mitosis phase, cyclin concentration and CDK activity is seen to be higher than the interphases. Okay, this is the cyclin expression cycle. at the G1 phase, it starts talking about the cyclin E first. So in the G1 phase, it starts progressing and it reaches to the high peak and then it starts declining. And what about cyclin A? That very moment of the G1 phase, it starts progressing and it reaches at the high peak of G2 and then it starts declining and reaches to the mitosis phase. Okay, and talk about the cyclin B, then it starts progressing from the S phase, that is synthesis phase, and reaches its high peak at the G2M phase transition. Okay, and then it starts declining and ends up to the mitosis phase. Okay, this is the representation. This is G0 phase, then it progresses to the G1, S, G2 and M. CDK4, 6 and cyclin D, it performs action from G0 to G1 stage, and thus 2N or diploid cell is formed, and then from G1 to S phase, CDK2 and cyclin E come into the scene. Okay, and from S phase to G2 phase, CDK2 and cyclin A, they come into scene and help in the genome duplication, and thus it forms from 2N to 4N. Okay, and then from G2 to M, CDK1 and cyclin A come into action, and thus it forms 4 higher degree of compaction chromosome. Okay, and then from M phase to G0 phase, then it forms you can say that it segregates chromosomes and it segregates or divides cell also. So CDK1 and cyclin B, they regulate all these activities. Okay, this is a cyclin-dependent kinase, that is CDK. CDK and cyclin, they both fit like a lock and key fashion. Cyclin binds to the CDK, as you can see, and cyclin CDK complex is then phosphorylated for further action. Okay, as you can see that it got phosphorylated, and this activated CDK and cyclin complex phosphorylates another target protein. Okay, this is the target protein, so it phosphorylates the target protein also. Then it is seen that activated protein advances the cell cycle. This protein, it advances the cell cycle, and this way cell is regulated by these two proteins, that is CDK and cyclin. Okay, I hope it's clear to all of you. Point are cellular inspectors. At first glance, the orderly progression of the cell through the phases of the cell cycle may seem perfectly straightforward. When building a house, the walls are not erected until after the foundation has been laid. Exactly. Likewise, in the cell, mitosis does not begin until after the genetic material has been copied. Otherwise, the daughter cells would end up with less than a complete set of chromosomes and would probably die. So, in a cell cycle, just as in house building, certain steps need to be preceded, others in an orderly fashion for the process of work. How does the cell know when a step has been completed and it's time to move on to the next? The answer is that the cell has several molecular inspectors which are stationed at intervals called checkpoints throughout the cell cycle. These cellular inspectors, they function much like building inspectors too. If a step has been completed to their satisfaction, they give the OK to move forward. If not, they halt progress until the cellular construction workers finish the task. There are three major checkpoints in the cell cycle. One between G1 and S phase, okay, G1 and S phase, and other is between G2 and mitosis, and the one during the mitosis itself. The concept of points in the cell cycle was first introduced by Ted Wienert of the University of Arizona in Tucson. Okay. In experiments with yeast cells, Wienert and Hartwell showed that a protein called RAD9 is a part of cell cycle checkpoint. Normal cells will stop and repair any damage to their DNA before embarking upon mitosis. Cells that lack RAD9, however, ignore the damage and proceed toward mitosis with catastrophic consequences. Having inherited damaged DNA, the daughter cells invariably die. Since these discoveries were made, other checkpoint genes have been identified in many kinds of cells, including human cells. Okay, Hartwell scientist has identified more than 100 genes that help control the cell cycle, and in recognition of the importance of these discoveries, he shared Nobel Prize in the Physiology or Medicine in 2001. So in short, these are the checkpoints. This is a spindle assembly checkpoint. It checks for the chromosome attachment to the spindle, and this point is between G1 and S phase. It checks for cell size, nutrients, growth factors, DNA damage. Okay, and then this is a G2 checkpoint which checks for the cell size and the DNA duplication. Okay, checkpoints in mitosis checks that are all chromosome properly attached to the mitotic spindle. It pulls duplicated chromosomes apart. Okay, and thus it helps in entry towards the G1 phase, and it is a checkpoint which checks is environment favorable. This is known as G1 checkpoint. Okay, and then it helps it into moving towards the S phase, that is synthesis phase, and this is G2 checkpoint. It checks is all DNA replicated, is all DNA damaged repaired, and then it enters into the mitosis phase. Okay, I'm again repeating, how does the cell know when a step has been completed and it's time to move on to the next? The answer is that the cell has several molecular inspectors which are stationed at intervals called checkpoints throughout the cell cycle. These cellular inspectors function much like building inspectors do. If a step has been completed to their satisfaction, they're okay to move forward. If not, they halt the progress until cellular construction workers finish their task. Okay, this is the proper illustration how cell progresses to a stage such as interphase, phase, metaphase, anaphase, telophase, and mitosis. Okay, so this comes to an end. In the next section of my presentation, we'll be studying about the cell cycle. So till then, stay tuned and keep watching Edupedia Word videos. Thank you.